Recording also. Perfect. So today we have a special guest. Amin, he's gonna present his recent work about knowledge spaces in cerebellum area. Today's talk is kind of special, not just by the topic, because Amin and I and Ali here also used to be in the same labs, and it's very cheering to see certain fellows from similar background doing all these great things. I give it to you, Amin. Thank you.
Thank you so much for the introduction. And yeah, of course, it's great to see you guys. Uh, Let me start sharing my screen. Okay, good.
Um,
So
just one sec. Let me try to see if I can turn on the volume more. Um, yeah, that's gonna be a problem because we don't have that.
do you guys see the correct screen? Is it the presenter or the
It's the presenter. Um, we're gonna need
okay.
another adapter for that.
There's one in the small meeting room.
Yeah, can you bring that? Sorry. I'm just gonna switch the adapter really quick. Yeah. Yeah, that's the max. I guess. Can switch it. This guy. Sec.
Can you guys also verify that you can see my cursor too on the
Yeah, I
screen?
can see your cursor.
Great, beautiful.
Okay. Let me see if it's working now. Speaker. Okay. Can you try talking now, please?
Yeah, can you hear me well?
Okay, no, this one probably. Can you try again, please?
Yeah, does it work now? Is it
Yeah.
better?
Awesome. Yeah.
Okay,
Yeah, let's get going. Thank you. Sorry about that.
right. Okay. Thank you for the invitation. I'm very excited to present the work that we've been doing for you guys and get feedback. So the title of our work is A Vector Calculus for Neural Computation in the Cerebellum. And before everything, I want to thank our fantastic group led by Reza, which has made this work possible. And I've learned a lot about like each individual in this lab, and it has been a great experience. And also, I want to thank our monkeys, who are basically the superstars of this work, who like dedicated their time for us to get the data and like figure out the brain. So let's start. So the main question that we're concerned with is the population coding. But cerebellum is nice because you basically have all these cell types and layered structures. You have the sole output, which is the Purkinje cells, and you have two types of inputs, the mossy fibers and the climbing fibers. And you have very well-known interneurons, and you know their connectivities. We know their connectivities. And we basically want to figure out that how the neural manifold with the constraints that are related to these connectivities relate to the behavioral map. So the question is pretty general question, but for a specific system that we have a lot of prior knowledge, and we want to try to see if that prior knowledge helps us understand the system better. So if you start with a little bit of history, a revolution in neuroscience has been like pushed forward with Hubel and Wiesel work. When they recorded from the visual cortex, they realized that there are neurons that caring about the specific features of the visual stimuli. Here, the direction of the visual stimuli, the direction of this line. And then the idea has been that maybe we can get from single cells to population. So around 6 years later, Georgopoulos had this idea, similar idea for the neurons in the motor cortex, that when the animal was reaching toward different directions in 3D, he was able to find that this neuron is caring about a specific direction. So you could have represent this neuron preferred direction or the potent vector with a vector. And then he could have made the population activity by adding these single neuron responses by the vector average of these neuron responses and basically showing that the population vector, the sum of these vectors would be toward the direction of the movement. But the backbone of this idea has been this assumption that single cell tuning curve is an estimator of its downstream effect, and there is no causal manipulation to do. So what happened 20 years later when Mark Churchland and Shenoy were recording from the same region, motor cortex, they've been very confused because they had these single cells that didn't show an obvious preferred direction, but also they were active way before the movement happens, during the preparatory period, that they showed that in the EMG, there is no muscle activity. So how can that be that these neurons are active during preparation and there's no EMG activity downstream? And the key idea, the key genius idea that they came up with was that the linear sum cancels during the preparation and forms the accurate motor commands for moving the muscles. So they've been looking for mapping the muscle space to the neural space using the weights W, which cancel during the preparation and give you the accurate EMGs during the execution of it. But as any other linear fitting, you have problems, you have challenges. The challenges is that you don't know what is your sampling bias in neural population. This can be cell types or the area of the recording. It can be what behavioral space are you using, what is the matrix M, and it can be even if your fit is unique or not. And what we've been trying here is that we've been trying to see if we can find the ground truth weight matrix because of the special properties that cerebellar cortex has. And as a role model, basically, David Robinson has been thinking about the eye movement system really beautifully. And he's been using the anatomical a priori as constraints of his models. And one of the interesting papers of his is that for the eye system, as probably many of you guys know, there are a set of muscles that are controlling the horizontal movements and vertical movements, and also the muscles enforce that too. For the vertical movements, it's slightly more complicated, but the horizontal movement, there is literally pairs of muscles. But what is interesting is that you have the motor neurons, which move the eye and hold the eye in the place, and you have premotor neurons that are building the saccade part, the movement part. But what was surprising for him was that these burst cells, these premotor neurons that were in charge of moving the eye horizontally, were active for vertical movement. So how can it be that I'm making a vertical saccade, but the neuron that is pushing the eye to the left and right is being active? And more interestingly, in the motor neurons, you don't see that. In the motor neurons, for the vertical movement, this activity is way less. So what he taught, what he modeled was that actually there are this pair of excitatory and inhibitory neurons that project to the motor neurons. His idea was that probably what is happening is that for the vertical saccades, the excitatory and inhibitory inputs are canceling each other, forming the null axis, and the potent axis would be a sharper response in time. So now why cerebellum? And basically, it is involved in multiple motor and cognitive tasks. The same structure, known neuron types, is handling all of these tasks. And I'm going to briefly talk about this structure. So the inputs to the cerebellar cortex comes through the mossy fibers from almost all over the brain. And then through the granule cells, parallel fibers give the input to the set of two types of MLIs, molecular layer interneurons, and the Purkinje cells. And the Purkinje cells are the sole output of the cerebellar cortex. So you kind of have a feed-forward inhibitory network with the skip connections that the output is sending the command to the deep cerebellar nucleus that later goes to the cerebellar cortex. More interestingly, these Purkinje cells get another set of input, climbing fibers, which are involved with the learning and potentiation of them, or depression. And the nice thing about them is that you can dissociate this learning signal from the output activity because these cells generate two types of spikes. We call them simple and complex spikes. Complex spikes in their waveform are very stereotypic. They're 1 Hertz activity. They have this calcium phase, slow calcium phase after the initial spike, downward spikes. And simple spikes in opposite are like baseline of 50 up until like 100 Hertz or even sometimes more. And these complex spikes are nice because they can basically have intrinsic perturbations for us. Because of biophysics, whenever a complex spike happens, the simple spikes pause for tens of milliseconds. And this is completely biophysical and can happen like anywhere during the behavior or before the behavior. In addition to that, they're involved in the potentiation and depression of the simplest spikes, meaning that if the Purkinje cell receives the parallel fiber input, and after a specific window of time, a climbing fiber input comes, the activity of the simplest spike, the synaptic connection of the parallel fiber to the P cell changes. So for the same movement, the output response of the simplest spike changes. So we have trial-to-trial changes in simplest spike induced by errors and teaching signal, which is the complex spike. So these two have been basically our torch to explore the cerebellar cortex, to move on the neural manifold and the simplest spike manifold to see how it maps to the behavior. So our goal is estimating the potent vector irrespective of neural tuning through intrinsic perturbations of the network. So what does the cerebellum do? And this is interesting because Gordon Holmes, a neurologist who was examining British soldiers during World War II, noticed that unfortunately, the British soldier's helmet had a flaw in the design, that the back of the head was not covered as opposed to the German helmet. And this led to, unfortunately, a lot of patients with visual cortex and cerebellar damage. And what he categorized the typical behavioral deficit of these cerebellar patients was dysmetric movements with the most obvious errors at the end of the movement. And you can see it in multiple modalities. So here is a patient with saccade dysmetria. As you can see here, when the target is presented, patient overshoots and again wants to correct and again overshoots. So you can imagine how hard it would be to just read an article. And similarly for reaching in the arm and hand movements. So the movement initiates, it's a smooth before reaching the end, but it's really struggling and frustrating to do the fine task of putting the cap on the pen. So how does the output look like? And people have recorded in multiple tasks, different regions of cerebellar cortex, how this Purkinje cells responds. And during the reaching, you can see that these Purkinje cells sometimes burst, they sometimes pause, decrease their activity, and sometimes mix. They're burster pausers, pauser bursters. This is true for whisking, this is true for licking, and for saccadic eye movements. In addition to that, you can see that the activities are way longer than the movement duration. So how can it be that the region that is really involved, seems to be involved from the patient studies in the accurate movement, endpoint accuracy, how can it have responses that are so heterogeneous and is lasting longer than the movement duration? So we use the marmosets as a sensory motor model to basically tackle this question. And as you can see, marmosets, beside being very cute, they have very nice gaze coordination, head and eye movements, and also tongue coordination. So their movements are very accurate. And we've been able to, with the lab mates, this is with the help of Alden and Paul, we've been able to set up the silicon probe recordings in the cerebellar cortex. And we've been able to train these monkeys to do thousands of trials in a single task to have neurons that we have thousands of trials for. And these trials entail licking, as you can see here, and the eye movements. So we've been able to record from 20 to 350 simultaneous neurons from multiple folia with up to 10,000 of licks and saccades per day. And the task that we did was a random corrective saccade task. The task starts with a fixation point. The monkey fixates. After a fixation time, a primary target is going to be presented. As soon as the monkey makes the saccade, the target jumps, inducing an error to one of the random 8 directions. We call the first saccade primary saccade, the second saccade secondary saccade, and the saccade to the fixation point, the center saccades. And these 3 saccades are necessary for the animal to get reward at the end of the task. And in order to prevent any type of learning, we basically randomize the primary and secondary target locations. But in addition to the reward relevant saccades, which would be these 3 types of saccades, we also analyze the reward relevant saccades thanks to the work of a master's student who worked with me, Jinyu, who modified the deep learning algorithm developed by the Hopkins lab to detect saccades for marmosets. So we are able to detect 10,000 to 20,000 saccades and microsaccades per session. So a little bit about cerebellar cortex and how our recordings are. The way that we record is that we came up with this more posterior burr hole to be able to put the neuropixel 10 millimeters inside the cerebellar cortex, in addition to remaining in the posterior vermis, which is the oculomotor vermis, the lobule 7, for the whole depth of the probe to be in the related eye-related regions. And for these monkeys, because of their size, 4 millimeter means multiple folia of recording. And here you can see the structure of the cerebellar cortex. So here are the molecular layer interneurons. You can see the Purkinje layer here with the Purkinje cells with larger cell bodies, and you can see the granular layer. And from in a slice studies, the nice thing about Purkinje cells is that these are really large cells, and the shape of the spike, the shape of the complex and simplest spikes tells you where on the cell you are. So a downward slow complex spike is observed in the dendritic tree, and the sharp sodium phase followed by a calcium phase of a spike is observed in the soma and the axon of the cell. So, and because the dendritic tree is in the molecular layer, and the soma is in the Purkinje layer, and the axon is in the granular layer, you basically can tell the polarity of the cell and the layer structure. I hope you guys can hear the sound, but here is an example recording. And you can see a Purkinje cell. The simplest spikes are the high frequency downward spikes. They sound like rain. And the complex spikes are 1 hertz sparse spikes, which sound like thunders. So the complex spikes, you can see they are upward, and simple spikes are down. And the usual recording looks like this. This is from 5 layers. Each of the dots are basically a neuron, and we have done cell type identification. I'm going to go through the cell type identification in more details as we move on. But for now, you can see the simple and complex spikes with downward shape for the simplest spike on the up. And as you go to the bottom, you have the sodium phase. So you already know the polarity that the molecular layer is on the top. We have MLI1 and MLI2s here. And the granular layer, we have the mossy fibers. For the next layer, it flips, as you expect from the anatomy, because the folias fold over each other. And for the next layer, it again flips and again flips and again flips. So how does the P-cell simplest spike modulation look like? So I'm showing you the responses of some example Purkinje cells for different directions of the movement that are concatenated here. And in yellow, you see basically the saccade velocity profile. So you see the movement duration. Some of these Purkinje cells, as shown before, pause. Some of them burst, but they burst and pause towards all directions. And also their modulation duration is way longer than the movement duration, as you can see. But this is puzzling because stimulation studies, both electrical and optogenetic stimulations, has shown that basically stimulating the Purkinje cells is going to push the eye toward a specific direction, no matter what is the direction of the movement. So if you have rightward movement, it's going to be pushed to the left. If you have leftward movements, it's actually going to be more hypermetric, and it's going to be pushed to the left again. So P-cell stimulation pulls the eye toward a specific direction, which is potentially our potent axis. So how can the responses be so heterogeneous and toward all directions? So this is our first puzzle. How are P-cells involved in endpoint control despite the fact that they're active long after the movement ends? And as I showed you earlier, complex spikes, the teaching signal has been very helpful for our lab to basically explore this problem. So how the complex spike responds is that they usually have a preferred visual-motor direction. They respond to both visual and motor. And as here you can see for leftward saccades, this example complex spike burst. And for the opposite direction, it has a pause, and it has a 1 hertz baseline activity. So we can represent this with a vector with the angle, which is theta CS, which is the CS on angle, preferred direction of the complex spike climbing fiber. And we can have a sharpness of tuning, which is the amplitude of this vector. And if we align all the 520 complex spikes that we recorded to the CS on direction, you can see there's a visual peak and there's a motor peak. When aligned to motor, you can see the build-up activity and the motor response. And as you go to the plus 90 direction, you're almost at the baseline. And for the opposite direction, you get a pause. So they're pretty homogeneous, and they're encoding a direction. And in addition to that, the sharpness of tuning changes. So you have cells that are very sharp, very much discriminate between the two directions. You have the cells that are medium, and you have the cells that are less responsive. But no matter the sharpness of tuning, in their perpendicular axis, they're blind to the errors. They're blind to the visual or motor events. So now let's use the complex spikes to have a hypothesis about what the Purkinje cells are doing. And we're going to do that, as we discussed, by the pause that they're causing. So we discussed complex spikes can suppress simple spike during the saccade, and this suppression is just biophysical. So the complex spike, whenever it happens, simple spikes are pausing, and this sometimes can happen during the movement. If we grab those trials that a complex spike happened during the movement, what we're going to see is that simple spike, instead of bursting, is pausing. Actually, you can think of it as a perturbation. And what is the consequence of how does it affect the behavior? What we observe, this is the work of Solomon Muller, is that the eye gets pushed toward the CS on direction with almost no components in the perpendicular direction. In addition to that, the work of David Hirschfeld showed that if you have a complex spike in the previous trial, the simple spikes goes through LTD, depression. In the next trial, it's going to respond less. And this smaller burst or like more pause is going to again lead to, in that trial, for the eye to be pushed toward the CS1 direction. So the idea is that this Purkinje cell has projections downstream to the motor neurons from anatomical studies, and the weights by which it projects downstream can be estimated by the CS on direction. And we have the negative sign because both the pause and the LTD are a negative effect on the simple spike, or a pause in the simple spike. So the idea is that the Purkinje cell pushes the eye towards CS on direction if it reduces its activity. So CS1 is an unbiased estimator of the downstream potent axis of the Purkinje cell. And we further push this idea forward by the fact that we have the complex spikes that have different sharpness of tuning. And is the complex spike vector only predicting the angle, or does it also information about the amplitude, the amount of the perturbation? And what we observed was that actually, if the complex spike is sharper, if you have a sharper teacher, your downstream is also affecting larger, meaning that the Purkinje cells that have sharper complex spikes push the eye more toward the CS1 direction, and the ones that have less sharp tuning push the eye less toward the CS1 direction. So we're going to update our weights to have the rho term, which is the sharpness of tuning in them too. So this is a similar problem to motor cortex, with the difference that we have a prior knowledge about the weights. So in that case, we had matrix M, which was the EMG activity, and we had N, which was the neural activity. But here we have the W from a priori knowledge of the intrinsic perturbations, and we have N, which is the neural activity. So we can basically ask, what is the downstream command N? But before doing that, we need to show if this relationship is really linear. Does these neurons add to each other, which is the idea of superposition? So do PISA responses add or cancel each other, meaning that if I perturb two of them, is the perturbation, is the eye going to be pushed toward a vector sum of the two? And second is that, is the CS on really the potent axis, and CS plus minus 90 would be the null axis, meaning that does the activity of these neurons cancel each other in the plus minus 90 direction and add up in some sense in the potent axis? So let's start with the superposition. So as I mentioned, the downstream effect of let's consider the downstream effect of two simultaneous Purkinje cells. The Purkinje cell 1 has the potent vector rho1 theta 1 that is coming from its complex spike tuning. And Purkinje cell 2 comes from rho2 theta 2. And then we show that the algebraic sum of these two vectors basically would be related to the ratio rho2 over rho1 and theta 2 minus theta 1, which we call it delta theta. So what we do is that, and this is, Hisham has helped with this analysis a lot in the lab, that because we have neuropixel recordings and we're recording from multiple folia, we have these cells that have difference in their preferred tuning. So if they simultaneously get suppressed by their complex spikes, what is the consequent move? And as you expect, we have more of the similar tuning just because the cells that are close to each other have similar tunings and similar potent vectors. But we have the vectors that are more perpendicular. So if the two vectors align with each other, the eye is going to be perturbed even more. And if the second cell is more upward, the eye gets pushed upward, so you can see diagonal. And if it is downward, it gets pushed downward. So you have kind of the vector sum is predicting what would be the behavioral consequence of the suppression of the two simplex spikes. So we can roughly add them. So now let's add them, because what we can consider is that we have a pseudo population of Purkinje cells. And we know that for a rightward movement, the left and right Purkinje cells, the Purkinje cells that care about right and left, are going to be the potent Purkinje cells. And we know from the weights that the rightward Purkinje cells are going to be negative because of what the weights predict, and the leftward are going to be positive. So when we look at these responses in our neurons, this is the Purkinje cell response to the left, which would be a positive effect, and this is the Purkinje cell response to the right. And we separate the Purkinje cells to bursters and pausers. And what you see is that when you subtract the left from right, as expected by the weights that were found, we get something, some activity that is sharper and is more time locked to the behavior. I'm going to show it in more details, but it's this burst pause effect. And if we do it for the pausers, although the pausers do not show any burst here, just it's the differential in the activity, you see the same pattern, the same up-down response. What about the null? So for the null would be the Purkinje cells that are caring about up and down for a rightward movement. And we see a symmetry in both bursters and pausers that cancel each other out. And basically, nothing is going to go to the downstream motor neurons. So the idea, and for the diagonal Purkinje cells, we get somewhere in between for both bursters and pausers. So the idea is that although for the rightward movement, the Purkinje cells that are caring about up and down, which are the null axis, are active, their activity cancels each other out and do not let the eye to get moved in an irrelevant direction. So to summarize, CS on reveals the potent axis. Px of t is projection on the CS on. And CS plus 90, which is this PYT, we see the complete cancellation because of the symmetry. Bursters and pausers show a similar projected response, although the responses initially are very different from each other. And P cell population pause here is time locked to the deceleration phase of saccade, and the pause ends at the end of the saccade, near the end of the movement. So now we're going to zoom out, and we're going to look at the cerebellar cortex as more of a network, because we know that there are these microzones in the cerebellum that are roughly around, as illustrated here, like around 100 micrometer, which entails multiple Purkinje cells. And the idea is that these Purkinje cells are receiving the same climbing fibers, so technically the same potent vectors. And we have MLIs as well, so how the interneurons are, what are the computation that is happening in the interneurons. But we don't have access to basically these nice maps because we have just EFIS recordings. But roughly in there, we can see that the Purkinje cells, these arrows are basically representing the CS on. You can see that in the layers, the cells that are closer to each other have similar arrows. Like this last basically region is not that eye related, and none of them are related. This region is more down and left, and this region is more up and left. So in order to find these compartments, we define cliques, which are within microzones. And the way that we did it was using the statistical spike interactions between these cells. So again, to just go through the network once again, we have MLI2s that suppress MLI1s, and we have the MLI1s which are suppressing the Purkinje cells. And in these recordings, we can observe these interactions by jittering the cross correlograms. So we can have jitter-corrected cross correlations, and we see that the simplest spikes have 0 millisecond synchrony with each other. MLI1s have 0 millisecond synchrony with each other. MLI1 suppress simplest spikes within 1 millisecond with a rebound, and MLI2 suppress the MLI1s, and MLI2s actually disinhibit the simplest spikes later in like around 3 milliseconds. And what we can do is that we can get a causal window in this and form an adjacency matrix for all the neurons that we have, which is the pairwise interaction of them. And what we observe here, the first thing is that not all these cells are connected to each other, and the cells within a layer are more connected with each other. So we can define the cliques using a spectral clustering of the graph. And we see that between the cliques, the interactions illustrated in blue are significantly less than within a clique. And also we observe that Purkinje cells synchronize, red means synchronization, increased inactivity together. MLI synchronized with each other here, and MLI suppressed Purkinje cells as illustrated in blue, which means the negative interaction. And in addition to that, this is mostly anatomical, but the MLI type 2s, we observe that are getting the spillover from the complex spikes, but MLI 1s don't, which is consistent with the previous studies of Alt and Heuser. So puzzle 2 for us is that can we generalize the potent space to other cell types with the help of this clique? So what we do is that the first step is that showing that really the cliques are within microsomes. And what the microsomes meant was that if the CS on, if the complex spike preferred direction is shared between them. And we are able to show that. So within the clique, the CS on angle difference is significantly smaller than between clique. And it's within like 22 degrees, which is pretty acceptable. So now we align the molecular layer interneuron responses to saccades to the clique preferred direction, which would be the Purkinje cell in that clique that has a complex spike, has the preferred direction, and we align the molecular layer interneuron activities to those directions of saccade. Was there a question?
Yes, there is a question.
Just a quick question. So, so the preferred direction in the CS is the same, but I mean, you could establish whether different Purkinje cells get the same climbing fiber input, right? To what degree was that true?
Yes, yes. So what we observe here is that Purkinje cells that are within a clique, that we have the complex spikes where we have the climbing fibers for, are sharing the same direction and same properties.
Okay, so yeah, I mean the two levels, right? They could share two different climbing fibers that have the same preferred direction, or they could share the same climbing fiber, and you can distinguish them. So, but,
Yeah,
but if they share direction, they also share the same climbing fiber, you're saying? Or?
That is more rare. Like getting the cells that exactly share the same climbing fiber is more rare. We have those, but that is not the norm. Okay. These are different climbing fibers that are sharing same properties, and they synchronize.
Okay.
But they're not the same climbing fibers. And we know if they're the same climbing fibers by having two simple spikes being suppressed by the same climbing fiber.
Okay.
But yeah, great question. Thank you. So, but to our surprise, when you look at the MLI ones, you really don't see that much difference between different directions of the movement, even aligned to the potent axis. And the same for the MLI twos. And more importantly, you don't see that functionally MLI twos do not seem to be suppressing the MLI ones as you expect from the anatomical structure. Probably is because you have the parallel fibers driving both of them positively. So what we do is that we project the MLI ones and MLI twos on the same potent vector to see how the projected responses look like. So as we discussed, the Purkinje cells show this burst pause pattern for both bursters and pausers. So I merged the bursters and pausers together here with almost no activity in the null direction. And interestingly, MLI1's differential response is opposite of the Purkinje cells. And if we plot it in the phase plot, you see the negative correlation between the two responses. And again, for MLI2s, if we project, we see that MLI2s are actually in phase with the Purkinje cells and opposite to the MLI1s, as again, you expect from the anatomical connectivity in the feed-forward inhibition network. So the idea is that interneurons seem to inherit the vector space of the Purkinje cells, and we are able to reveal the code, although their raw responses do not seem to be that different, and they're bursting toward all directions. But the subtle difference in their delays for different directions is giving us this differential response. So puzzle 3, what is being computed by the network? So what does this burst pause in the Purkinje cells mean? And in order to answer that question, we're going to look outside of the cerebellum a little bit to see what are the inputs that are coming to the system. So we know that for the saccade generation, superior colliculus is very much involved, using the inputs that it's getting from the cortex and basal ganglia. And superior colliculus sends, and these are from anatomical studies, we noticed, that sends the sensory goal to the cerebellum through a structure called an RTP. And in addition to that, superior colliculus derives the burst generators, the premotor neurons, and also cerebellum gets input from these motor commands. So we know that there are these two types of mossy fibers that are going to the cerebellum at least. And the cerebellum talks to the burst generator to basically do the correction that is required to do. So, and when we look at the patients, they have problems with accurately stopping the movement on the target, which makes a lot of sense to hypothesize that this pause really is pushing the eye back to land it correctly on the target. But how are we going to make sure that that's the case? And what we are going to do is basically, we think that we have the neural manifold of interest by the projected space and the potent vectors. So now we're going to walk on the behavioral manifold of interest, which is the saccade properties, to see what are these regions are corresponding to the neural manifold, how the neural manifold change if we change the saccade kinematic properties. And one of the interesting things that we have in our data is that intuitively we know that reward alters how we move. When we are meeting a friend, we walk faster to reach sooner. And this is kind of what we see in our monkeys too. So for reward relevant saccades, you look at the saccade velocity profiles, and they're shorter in duration, they have higher velocities, and their deceleration phases especially is smaller in comparison to the same reward irrelevant saccades that are being made with the similar amplitudes. So we're going to look at how does reward in these two groups of movements are affecting the inputs, the mossy fibers that we can record from, which I'm going to say how we're detecting them, and how does it alter the activity of the computational units that we have, which would be the cliques, the MLIs, and Purkinje cells, as we discussed. So in order to detect the mossy fibers, and people have been doing this since 1992, Otsuka and Noda, basically mossy fibers have various stereotypic spike waveforms in the granular layer. And thanks to the complex spike, we have the layer information. So the mossy fibers in the literature usually either have a short duration, a sharp spike, or they have this M phase with a negative after wave. That Mike Heuser lab has recently shown that negative after wave actually is related to the postsynaptic response of the granule cells. And by blocking the granule cell synapses, they're able to remove that negative after wave. And what we did was that we used neuropixel ultra recordings, which are basically the neuropixels probes, but with very higher resolution, to show that a single Rosetta structure is showing both the M shape with negative after wave and the fastest spiking waveform. So it is from the same Rosetta structure. It just depends where your probe is with respect to the rosette. And then what we were able to do was that we were able to, in the granular layer, look at our spike waveforms in a spatiotemporal maps, which would be one of the columns of these recordings, and detect the mossy fibers. And you see in the UMAP space, we have two groups of the mossy fibers that are either fastest spiking or the M shape with a negative after wave, versus the granular layer interneurons, which are larger in size and wider in the duration of their responses. So we were able to detect the mossy fibers. And out of all the groups of the mossy fibers, two groups that were interesting for us were the copy of the motor command, which are basically look like the premotor neurons. For reward, this neuron, this is an example neuron that is caring more about downward saccades, and this is being by the duration of saccade. And as you can see, it is very time locked to the velocity profile, and actually the rate looks like a lot like the velocity profile of the downward movements. And the number of spikes is very much correlated with the amplitude of them. And for the reward irrelevant saccades, the same rule holds. So the premotor neuron doesn't care if you're making a reward relevant movement or reward irrelevant movement. Your burst durations are going to be longer because your velocity duration is longer, but otherwise the encoding is the same. But as opposed to that, unlike that, you have goal mossy fibers that are potentially the signal that is coming from the colliculus, from the NRTP. You have these neurons that have build-up activity aligned to the saccade onset. This cell cares about left up. They have amplitude tuning. They care about a specific amplitude. And for reward irrelevant saccade, they have more muted responses. So it's not only about kinematic, but they also convey the goal with respect to the context of how much you care about this movement. And we're able to use this features basically to divide all our cells to a state and goal. And we don't use the preferred directions, but what we observe is actually our state MOSI fibers are in the motor coordinates, are vertical and horizontal, but our goal MOSI fibers are actually in the collicular coordinate. They can be diagonal, as you see in the colliculus. In addition to that, the state MOSI fibers have wider angular tuning curves as opposed to the goal MOSI fibers having sharper responses. And as I showed, if you group your goal MOSI fibers by low amplitude preference, medium amplitude preference, and high amplitude preference, you're going to see that the responses are more muted for the irrelevant group than the reward relevant group. But for the state MOSI fibers, there's no difference. And here I'm just showing that the linear relationship between the saccade amplitude and number of saccades is no different for the reward relevant and irrelevant saccades. But for the goal MOSI fibers, there's an obvious difference in the tuning and the number of spikes generated for reward relevant and irrelevant saccades. So now that we know that the input is different, let's answer puzzle 3, that how does it affect the interneurons in the projected space and the Purkinje cell outputs. So for the reward relevant, what we observe is that no matter what peak velocity you have, the zero crossing that we discussed that is exactly at deceleration onset happens for the Purkinje cells no matter what. Also, the MLI1s reached a similar peak in the projected space during the deceleration. And MLI1s and Purkinje cells remain on the similar manifold, with the deceleration onset being in a specific location of the zero crossing on this manifold. But for the reward irrelevant saccades, the zero crossing is happening way later or not at all. And the MLI1s are not reaching the same peak. But the MLI1s and Purkinje cells still remain on a similar manifold. But the deceleration onset in time is not anymore encoding a specific part of this manifold. So probably the network is not involved as much anymore. In order to show that further, we bin the saccades by their acceleration duration, which is basically from the saccade onset to deceleration onset, is unknown, and the saccades can vary in that respect. And we show that for most of the groups of the saccades that we have, from like 7 millisecond acceleration duration up until 14, 15 millisecond acceleration duration, which is almost twice more, the zero crossing remains time locked to the deceleration onset, except for these very long acceleration durations we realize that are like these more weird shaped saccades, that a pause happens during the saccade. So the velocity stops and then goes again, as if the saccade stops and goes again. And we call these double step saccades, that the zero crossing gets pushed back more. But for all cases, again, the MLI1s reached a similar peak, but not for the irrelevant saccades. For the irrelevant saccades, for the similar acceleration durations, you get less than half peak for the MLI1s in the projected space, and the zero crossing is not happening anymore at the deceleration onset. And for all these scenarios, again, MLI1s and Purkinje cells remain on a similar manifold as you expect, that is probably potentially restricted because of their synaptic connections, which is an inhibitory network. So our conclusion here is that P cell signal when the saccade should begin decelerating, but only if the MOSI fibers provide location of the target. So in order to summarize, I'm going a little bit over time, sorry about that. We tried to address 3 puzzles. Puzzle 1 was that how are P cells involved in endpoint control despite the fact that they are active long after the movement? And what we realized was that if we know what is the downstream effect that they have on the behavior, and if we use that as their weight, we can find a projected space that has a common encoding both for bursters and pausers, which is basically caused by this delay that is for different directions. Our second puzzle was that can we generalize this for the interneurons that we have, that we know the connectivity form, and does it make sense that they're suppressing each other functionally? And the answer was yes. Forming the cliques of interconnected cells enabled us to reveal the code for the interneurons as well, and it anatomically made sense. And our third puzzle was that what is being computed by the cerebellar cortex, which we believe is this timing, that when you stop the eye, if you care about the movement to be accurate. Thank you so much for your attention, and I'm looking forward to have your questions and feedbacks.
Thank you. Okay, we can start with questions.
I have a question actually. I might miss that, but I was wondering if you can see any special
Excuse me,
organization.
I cannot hear you clearly.
Yeah.
Is there anybody that's sorry?
Hi, Amin. Thanks for a great talk. I was actually wondering, maybe I missed that and you said that already, but can you spot a special organization for the encoding of the different movement directions?
Yeah.
Yeah.
So that great question. That has been actually I think I have a slide for this. Sorry, give me a second. Yeah, here the work of David Hirschfeld, and we also observe it, it is in like the previous papers too, is that the CS on direction is very much dependent on if you're in the right part of the vermis or left side of the vermis. So there is like colliculus, you have a spatial map that the cells closer to each other, and also you know it from the microzones, that the cells that are closer to each other are getting the same inferior olive inputs and similar CS on directions, which we showed that is correlated with their downstream effect. Did I answer your question?
Yeah, thank you.
I mean, I have a question for the clustering or the clique finding and the microzones. What's your approach for the neuropixel? Are you basically inserting perpendicular to the fovea direction, or is your approach like oblique, or you know, where do you approach?
Yeah. So in order to remain in the same region, so this is lobule 7, and it took a little bit of risk to go this posterior because the confluence of sinuses here, which you don't want to hit with the probe. So what we did was that we used the T2 MRIs, and we planned the trajectory that we missed that for sure. So we put a margin that that's not going to happen. But otherwise, what we wanted to be as parallel as possible to the lobule 7, that as the z-axis goes inside, we remain on lobule 7. And the more posterior we did our vehicles and our implants, the more successful we were.
I'm not quite sure if I understand right. So perpendicular to the lobulus, define. So there's the direction of parallel fibers and the fovea, the folding. Are you exactly 90 degrees to that, or are you going? When you say parallel to lobulus, what does it mean?
Yeah. So it means that, so basically this is the lobule 7. So if you see my cursor, we try to be perpendicular when we enter the surface of the cerebellum, but we try to remain in the same region as our trajectory goes forward. Does it make sense?
Kind of, I guess. But you're going through different folia as you showed, meaning you know that, I mean, the microzone organization is usually parallel to the folia. So you don't have a recording right now where you're in the same folium, but you go basically along the folium.
Yeah, we don't try to remain on the same microzone. We want to just remain in the same, and I honestly, I'm not sure what is the microzone in the vermis, because many of those in our studies are in mice, and also the vermis doesn't seem to have that much different zebrin. So I'm not sure how that organization actually is going to be, and that is partially why we do the click analysis to find the cells that are interconnected and then show that they're sharing the same CS on, which is the meaning of being in the same microzone. But what we try actually is that we try just to remain in the eye-related regions, which is lobule 7. We don't try to, so actually we try to be more diagonal to cross the vermis to get opposite CSOs. Does it make sense? So we don't try to remain on the same side or anything. We try to like basically get the more diverse population with different CSO directions.
Okay.
I have one question. Maybe it's not a question, and I don't really understand what's going on the cerebellum at all. But the existence of the kind of Boston and pause mean that some kind of constant driving force on the motor neuron is kind of ongoing, right? And it's the cerebellum is kind of modulated. So what's kind of the direction the animal would go on that way? Is that right? So is that kind of the constant kind of driving force kind of other source than the cerebellum, or the cerebellum circuit is antagonized all the way, always, and it kind of sometimes pauses and sometimes bursting to kind of to move that eye to the specific direction. So I just want to kind of make sure that what's going on there.
So if I understood correctly, you're asking about the bursters and pausers and what they do. And it's interesting to actually look at this because both in macaques and marmosets, in almost all the data that we recorded from the 4 monkeys and David Hersfield results in macaques from Robi and Yoshiko's lab, is basically that the pausers and bursters are almost 50-50. Pausers usually have like a little bit more, like I have 125 pausers and 101 bursters. And it's interesting to think about where this cancellation is happening. And what we think right now is that the bursters and pausers can even be in the same click and the same side. Okay, so what we think is that in the cortex, you have bursters and pausers. And if you average these two with each other, what you're going to see is that like the Nature paper, the 2015 paper of the David Hersfield, you're going to see that effectively you get a sharper burst in the right side and a later burst in the left side, which is again more time locked to the movement. So you have a kind of knowledge space cancellation in time through your pausers and bursters. And fastigial response like this too. So if you record from fastigial nucleus, this is the effective inhibitory input to the fastigial. In the fastigial, you see interestingly that on the right side, technically, you get a later burst, and in the left side, you get an earlier burst. And the burst is sharper than what you see in the cortex, in the Purkinje cells. But again, in the fastigial, you get responses for the other direction. You get the responses for the upward directions. And that is also predicted by the model because the Purkinje cells are active. So knowledge space in time is partially canceled from the Purkinje cells to the fastigial, but not completely. So we believe that the cancellation potentially is happening in the motor neurons and premotor neurons by the EBN and IBNs. Does that answer the question?
Yeah, thanks so much. Yeah.
Of course. Great question. Thank you.
Okay, any more questions? Okay, I guess not. Then okay. So thank you, Amin, for presenting your work. And yeah, have a great day.
You too. Thank you
Bye
so
bye.
much. Have a good time.